Hello, everyone. Good morning. Welcome to Lunch with a Scientist. I'm Leah from Headwaters Science Institute, and I'm very excited to welcome you to another episode where you will get to meet a professional scientist doing some really interesting work, this time on frogs. So, in a minute, I'm going to pull up Jordan Gass. She was born and raised in New Mexico and completed her undergraduate degree at New Mexico State University, double majoring in biology and microbiology. While in college there, she discovered a passion for disease ecology and decided to pursue a master's degree at the University of Nevada, Reno. So she is now studying the host-pathogen interactions of amphibians with a pathogenic fungus. And if that sounds like a mouthful to you, well, you'll get to hear all about it and more today with Jordan. So I'm excited to welcome her. Hi.
Hi, how are you?
I'm great. How are you?
I'm doing well, thanks.
Well, we're super excited. As we said off screen, we've had quite a few frog scientists talk to our audience here. So we're connecting more of the pieces about frogs and getting to learn more today.
Yeah, definitely.
All right, let's go ahead and pull in your screen share.
Okay.
And we'll let you get going.
Okay, great.
Yeah, you're all set.
Okay, sounds good. So I'm really excited to be talking to you guys today about my research looking at the innate immune system of this cute guy right here, the Panamanian Golden frog. And I'll begin with just a little review on some of my background that Leah already touched on. I grew up in Las Cruces, New Mexico, where I went to New Mexico State, and I got degrees in biology and microbiology. And while as an undergrad, I had the opportunity to work in a biology lab that worked with dengue virus. Dengue virus is closely related to some other viruses you may have heard of, like yellow fever virus, Zika, and West Nile. And dengue is transmitted between human hosts through mosquito vectors. So some of the research I was doing while working in this lab was looking at the fitness consequences of evolution of this virus in both its human host through tissue culture experiments and in its mosquito host. So this experience just got me interested in disease in general, but it also specifically got me interested in how complex and dynamic host-pathogen interactions can be. And also my senior year of undergrad, I had the opportunity to go to Costa Rica on a field course. And here we got to visit both the rainforest and the tropical dry forest. And this really cemented a passion for ecology and conservation biology. And so moving forward, I became very interested in the field of disease ecology, which is the study of hosts and their pathogens in their shared environment. And when we think about a host, we might commonly think about humans and the pathogens that affect us, like viruses and bacteria. But because nearly every animal has pathogens that affect it, the field of disease ecology is extremely widespread. So it can encompass either studying dengue virus transmission between humans and mosquitoes in a large city, but it also can involve studying the pathogens of amphibians in a Panamanian rainforest. So with that in mind, I decided to pursue a master's degree at the University of Nevada, Reno, where I'm in the Evolution, Ecology, and Conservation Biology program. And here I've combined all of my interests. So I'm looking at the host pathogen interactions using an amphibian and a fungal pathogen study system to study those interactions. And so for this talk, we'll begin by talking about the fungus disease study or disease system that I study. And then we're going to zoom in a little more closely on the history of this fungus in Panama, which is where the frog I study, the Panamanian golden frog, is from. And then we're going to end by talking about my master's project and looking at the results I have so far. So let's begin by just looking at amphibians in general. So this graph is showing the distribution of global amphibian species richness, and we can see fewer number of species represented by light green, moving up a gradient where dark blue is representing a lot of species. And what's immediately noticeable is just how widespread amphibians are. So they inhabit every continent except Antarctica, and the greatest species diversity is clearly in Central and South America, as well as along the equator in Africa. But unfortunately, amphibians are one of many groups of organisms that are experiencing precipitous declines. So this graph is showing the number of threatened amphibian species with a similar trend where lighter yellow represents few species, moving to dark red representing many threatened species. And we can see there's several kind of hot spots for threatened species in South Asia, Australia, and then again in Central and South America, where we saw the greatest diversity of amphibians as well. And there's a number of reasons why amphibians are declining, including habitat loss, invasive species, climate change, pollution, and disease. And while all of these reasons are important and still prevalent, in the last few decades, disease has really emerged as a dominant driver of amphibian declines. And the pathogen behind these disease-induced declines is a fungus called Batrachochytrium dendrobatitis, or BD. And that's a huge mouthful, so I'm just going to be calling it BD for the rest of the talk. And BD is a ubiquitous fungus that's found in both the soil and water, and it infects amphibian skin. So this figure here is showing the life cycle of BD as it infects an amphibian. So BD begins in this motile zoospore stage. At the top left corner of the figure, we can see it has that sort of tail, which is called a flagellum, which it uses to move through the environment until it finds an amphibian's skin, represented here by these purple kind of blobs of skin. And once it finds the skin, then it can encyst into that skin and move deeper down into the amphibian's skin layers, where it matures into what we call a sporangium. And once it's a sporangium, then it reproduces asexually to produce a lot of those tiny zoospores that we saw at the beginning. And once it's ready, then it moves back through towards the top of the amphibian's skin, and it releases those zoospores out to the environment. And I think what's made clear by this figure is how disruptive this process is for the amphibian skin. So we can see some of the skin being sloughed off and being clumped up in abnormal ways. And this is especially detrimental for amphibians because their skin is extremely important. Not only does it protect them from the environment like our skin does for us, but it also serves major physiological roles like ion transport and respiration. So if any of those roles are disruptive, it's pretty clear how this fungus can be extremely detrimental for frog health. So these are two pictures taken from the field where a BD outbreak had just occurred. And it's clear from these photos that this pathogen can be extremely detrimental and is a huge conservation concern for amphibian biologists. However, these kind of catastrophic die-offs are not the case for every amphibian that's infected with BD. So BD infection outcomes exist across a spectrum, where on one end we have highly resistant frogs that show really no signs of disease. And because of this, they're able to carry the fungus on their skin and potentially transmit it to new environments and new amphibians. And an example of an extremely resistant frog is this African clawed frog. And on the other end of the spectrum, we have amphibians that are extremely susceptible to disease. So they show major clinical symptoms like lethargy, that skin sloughing I was talking about earlier, and abnormal posture. And unfortunately for highly susceptible amphibians like this wood frog, the result of a BD infection is almost always death. But what makes this BD pathogen system really interesting is that BD doesn't just infect one resistant and one susceptible host. BD infects literally hundreds of different amphibians. So that's what's represented here in this figure. So if you look at the smallest circle in the middle, that's separating these amphibians out into order. So Anura represents frogs and toads, and that smaller piece of the pie, Caudata, represents salamanders. And then the second circle is separating the amphibians out by family. And then those tinier colored bars on the outside are individual frog species. And the color of the bar represents how severely they've declined because of this pathogen, with the dark blue representing that amphibian species has declined less than 20%, ranging all the way up where yellow is greater than 90, and red and dark red are presumed extinct or extinct. So it's had wide-ranging effects, and part of the reason why we know that BD infects all these different species is because BD has been spread all over the world. So here we can see the distribution of BD-induced declines. And it's clear from looking at this that some areas have been hit by BD much harder than others. So we can look at Central America, South America, and Brazil show much higher rates of amphibian decline than in other areas of the world where BD exists. And if we zoom into Central America, we can see that unfortunately the light blue bar represents about 80 species have experienced between 20 and 90% declines. We have about 60 that have experienced greater than 90, and 20 are presumed to be extinct because of BD. So clearly it has had a huge effect on Central America, and specifically in Panama as well, BD has been extremely detrimental. So Panama is a small country in Central America that's home to a huge diversity of plants and animals. So if you get to go to Panama, you might be lucky enough to see something like a jaguar, this black-footed spider monkey, or this keel-billed toucan. But one of the most revered animals in Panama is the Panamanian golden frog. So these guys are cherished by the people of Panama, and they're considered to be good luck, but they themselves have not been lucky in terms of their susceptibility to BD. And BD first entered Panama in the mid-90s through Costa Rica, and it's continued to move southeast through the country well into the mid-2000s. And luckily for us, the lab I work for right now, Jamie Voiles' lab, had already established several research stations in Panama before the outbreak of BD. So those research stations are represented here by the 1, 2, and 3. And this gave our lab the unique opportunity of being able to see how amphibian populations change, both before a BD outbreak, during a BD outbreak, and now over a decade after the outbreak of BD. And because of that strength of time, we were able to see certain patterns. So this figure is showing the percent species detected on the Y, and then moments of time on the X, and then each little bar on the graph represents a different species. So in terms of moment of time, we're looking at pre-disease. So that's the population size before BD had entered Panama. And then we're looking at the epizootic stage. So that's right when BD had been introduced and was causing declines. And then we're into the enzootic stage. And that's when a pathogen has been in an area for quite some time. So we can see immediately that from pre-disease to epizootic, as soon as BD was introduced, all these amphibian populations declined. But what's interesting is that 10 years after that initial introduction, in the enzootic stage, we see all those populations start to rebound. So that makes it clear that amphibians seem to be recovering from BD, but how are they doing this? So when we're thinking about how a host responds to a pathogen, we need to think about the host's immune system. So amphibians have extremely complicated and complex immune systems, just like humans, that are separated into both an innate and an adaptive branch. And the innate branch of the immune system is characterized by being very rapid to respond to an infection. And part of the speed is because this part of the immune system is not specific to any one pathogen, but responds to sort of general pathogen markers. And partly because of this, it generates no memory against these infections. So sort of key players in the innate immune system are cells like these macrophages, which are able to non-specifically target pathogens and then gobble them up and degrade them, and these skin secretions, which can degrade a wide variety of pathogens. And on the other hand, amphibians also have an adaptive immune response, which is, in contrast to the innate, much slower to initially respond. However, this side of the immune system is specific to pathogens, so it's able to target an individual pathogen, and it's also able to remember that pathogen. So upon second exposure, we have a much faster response. So some things associated with adaptive immunity, you may have heard of antibodies, which are able to specifically bind to certain parts of pathogens, and also T cells, which are able to target infected cells within the host and destroy them. But based on our previous research and other research in the field, it's been suggested that the innate immune system is more important for BD infection in amphibians, and our research specifically has pointed to skin secretions as being an important component of the immune system. And so the amphibian skin secretions are made of an amalgam of molecules, including the amphibian's natural microbiome, antibodies that are secreted onto the skin, and also antimicrobial peptides, which are small peptides that can degrade a wide variety of pathogens. And out of all of these, it's clear that amphibians really shine in the production of antimicrobial peptides. So this figure to the right is showing the number of antimicrobial peptides produced by a wide range of different organisms on the x-axis. And when compared to mammals, fish, and arthropods, amphibians go way above and beyond in terms of the number of antimicrobial peptides they produce. So all of this kind of comes together to indicate that skin secretions may be a particularly important immune defense in amphibians against this pathogen. And this is some of the preliminary work that was done in our lab to establish that. So in this first figure, we have inhibitory effectiveness on the y-axis. So in other words, that's how effectively the skin secretions inhibit BD. And on the x, we have different species of amphibians. And then the two bars here represent the skin secretions taken in the pre-disease part of time. So this was before BD had been introduced, and that's represented by light blue. And then we also have the skin secretions taken during the enzootic phase. So that's after BD has been there for many years, and that's represented in dark green. And you can see that the dark green, so the skin secretions in the enzootic time, are much better at inhibiting BD than the skin secretions from the same frogs taken in the pre-disease time. And the same trend is shown in panel B, where again, we're looking at inhibitory effectiveness on the Y. And this is comparing captive versus wild Atelopus frogs, where the captive frogs have not been exposed to BD, and the wild have been evolving along with BD for many years. And again, it's clear that the wild frogs have a much greater inhibitory effectiveness against BD. So all of this points towards the importance of skin secretions in protecting amphibians in general, but specifically in protecting them against this BD pathogen. So that led us in the lab to ask, well, what's the role of skin secretions in protecting the Panamanian golden frog from BD infection? So that takes us to my experiment. So we were curious what role the skin secretions played. And to get a sense of how important skin secretions are, we wanted to see what would happen if they were no longer there. So to eliminate skin secretions, we used an injection of a hormone called norepinephrine. And what norepinephrine does is it stimulates a stress response within the amphibians, which causes them to secrete all of the skin secretions they had on their skin and in their glands. And this norepinephrine stress response is dose dependent. So a high dose of norepinephrine will cause a high stress response in the frogs, which will cause them to secrete all of their skin secretions. But a lower dose of norepinephrine will cause a slightly lower stress response, and that will result in the frogs secreting some, but not all of their skin secretions. And to act as a control for this experiment, we also use saline injections. So the injection of saline will recapitulate the stress of receiving an injection without actually causing the stress response in the frogs. So frogs that receive a saline injection will be able to retain their complete skin secretion repertoire. So based on all of this, my hypothesis for this experiment was that the skin secretions would play an important role in the immune response of the Panamanian golden frog against BD. And based on that, the predictions I made were that the frogs with no skin secretion are going to be more susceptible to BD. And because of that, they may die faster from infection, they may carry a larger pathogen load, and they may show more severe signs of disease. So here's a schematic of the experimental design. So I broke the frogs up into 5 treatment groups. For group 1, these guys were given that high dose of norepinephrine. So remember, the high dose signifies a high stress response. So these guys secreted all of their skin secretions. So they no longer have that component of their immune system. And then we exposed these frogs to the pathogen. So we infected them with BD. Similarly, for group 2, we gave these guys the lower dose of norepinephrine, so they secreted some, but not all of their skin secretions, and then they were also infected with the pathogen. For group 3, these guys were given that shot of saline, so they were able to retain all of their skin secretions, and then they were also exposed to BD. So for groups 1 through 3, all of these frogs have been exposed to the pathogen, with the only difference between the groups being how many of their skin secretions they have. And then for group 4, this served as a negative control group. So they were also given an injection of saline, so they retained all of their skin secretions. And then these guys were exposed to heat-killed BD. So heat-killed BD is BD that's been placed in very hot water, so it's no longer alive, it's dead. So this BD cannot infect the frog and cause infection. However, because it's dead doesn't mean that the DNA within the pathogen has been degraded. That DNA is still intact. And so some of my future research is going to be looking at how much of the pathogen's DNA is on the frogs at different time points. And because the DNA of this heat-killed BD is still intact, these frogs may appear to be BD positive. And to account for that, we added in one more negative control group. These guys were given an injection of saline, so again, they kept all of their skin secretions intact, and then they were exposed to just pure media, so they were not infected with the pathogen. But for the purposes of the results I'm going to be showing you guys today, we can consider group 4 and group 5 as equivalent negative control groups. So the first thing I did to begin the experiment was I just took some diagnostic measurements of all of the frogs. So I began by weighing each of the frogs, as shown here on the left, and then I measured the frog's body length using this tool called calipers. I measured them from their snout to their vent to get their snout vent length. And then finally, I took diagnostic swabs of the frogs, as shown on the picture on the right. So I swabbed their stomachs, thighs, and hands and feet. And the purpose of those swabs is so that in the future, I can extract the pathogen DNA and quantify pathogen load. And the purpose of getting the frog's weight and length at the beginning of the experiment is so that I have a sense of what is their body condition at the beginning, because I'm going to monitor body condition over time. And after I got those initial measurements, then I depleted the skin secretions. So this is the same figure I showed you guys earlier. Just as a reminder, group 1 received that high dose of norepinephrine, so they fully secreted all of their skin secretions in water, which I was then able to collect. Group 2 received that lower dose of norepinephrine, so they only partially secreted their skin secretions. And then groups 3 through 5 received a saline injection, so they did not secrete any of their skin secretions. They maintained that aspect of their immune system. And after these depletions were completed, I was ready to infect the frogs with BD. So to grow out BD in the lab, I just grow the fungus on media in these tissue culture flasks, which is shown in that left photo. And then when I'm ready to collect them, I just collect all of these zoospores and dilute them to a set concentration. And that's to ensure that every frog was exposed to the same concentration of pathogen at the beginning of the experiment. And once I had that BD ready, then I placed frogs in their exposure containers. So that's shown in that middle picture. They're sitting in those tiny containers on the counter. And I had them sit in a bath of BD zoospores for 24 hours. And this little animation on the side is just a summary of what I did. So I took the BD zoospores, diluted them in media, and then poured those on the frogs and had them sit in it for 24 hours to establish infection. And then once a week until the end of the experiment, I repeated those measuring, weighing, and swabbing protocols I did at the beginning. And again, this is so I can get a sense of how are the frogs' body condition changing over time, and how is the pathogen load on the frogs changing over time. And with that, we can get into some of my results. So this first figure is looking at body condition at week 1. So this is before the infection started. How did the frogs start? How did their body condition compare to one another? And here we have body condition on the y-axis, and then the treatment groups on the x. And when you're interpreting these box plots, the letters at the top represent significant differences. So if the letter is the same, then two groups are not different from one another. If the letter is different, then they are significantly different. So we can see that groups 1 through 4 started off with a very similar body condition. There's no difference among groups. However, that last negative control group, group 5, started out being much smaller than the other frogs. And so this isn't a huge deal, but it's important to keep in mind when we look at how the body condition is changing over time. which is shown here in this figure. So again, we have body condition on the Y. This time we have time in weeks on the x-axis. And you'll note that that fifth negative control group, represented here in the light pink, is much lower than the other 4 groups, which is what we showed in that previous graph. But what's really important to take away from this figure is that both negative control groups, shown in the light pink and the light purple, maintain a relatively stable body condition over time. So their body condition isn't really changing. And this contrasts to groups 1 through 3, which were all exposed to BD. Their body condition is all decreasing over time. And the same type of thing is shown in this figure. So this is just showing, instead of body condition, it's change in body condition on the Y, where a greater change is a higher number and a lesser change is a lower number. And again, we see the same trend where our negative control groups, shown in the purple and pink, have a very low change in body condition. So their body condition doesn't change over time, whereas the 3 BD positive groups all have a high change in body condition. So this is clearly a result of BD infection, which is causing their body condition to decrease over time. But we're also interested in the role of skin secretions, right? So remember that this saline BD positive group, that light green, they maintained all of their skin secretions. So we would have thought that they would have been better able to maintain their body condition over time relative to that low dose and high dose treatment. But that's not what we see. We see that all those 3 groups change their body condition equivalently over time. But we weren't just looking at body condition, we also were interested in frog survival. So here we have mean survival time in days on the y-axis, and then the 3 exposed treatment groups on the x. And what you'll note is there seems to be a slight trend where the high dose BD positive group is surviving slightly longer than the low dose, which is surviving slightly longer than the saline. But if you recall, this is the opposite as our prediction. So we predicted that the saline BD group, because they retained all of their skin secretions, they would be better at surviving than these other groups. But that's not what we see. But it's important to keep in mind here as well that if we look up, we see that all 3 of these groups have the same letter, which means that they are not significantly different from one another. So our main takeaway from this graph is that all of the 3 BD exposed groups survive approximately the same amount of time. And another way of looking at survival is through a survival curve. So here we have percent survival on the Y and days on the X. And you can see that every treatment group begins at the top at 100% survival. And that pink line across the top represents the negative control groups, which retained 100% survival throughout the course of the experiment. And this makes sense, right? Because those negative control frogs were never exposed to the pathogen, so they shouldn't have died, and they didn't. And what we're really interested in is how quickly the high dose, low dose, and saline BD exposed groups are dying. And you can see here that by day 25, frogs in those groups begin to die. And by around day 40, they have around 50% survival. And then by the end of the experiment, around day 55, all the frogs in the exposed groups have died. And what we really care about is seeing if the saline BD positive group, so the group that maintain their skin secretions, are they dying any later than the frogs that lost them? And no, it looks like they're all dying at a very similar rate. So this implies that skin secretions don't really increase frog survival or frog health in terms of body condition in the Panamanian golden frog. And that suggests that these skin secretions might not be as important of an immune defense against BD, at least in these specific frogs. But in the summer, I'm hoping to do more research to investigate this. So the first thing I want to do is quantify the BD load on the frog. So those skin swabs I was taking, I want to actually extract DNA from those swabs and then use some molecular techniques to quantify how much BD load is on the frogs at certain time points. And what I'm expecting to see here is that potentially the frogs with skin secretions may have a lower BD load than the frogs without. And I also want to test the effectiveness of just the pure skin secretions that I collected from the frogs at inhibiting BD in vitro. So in vitro just means in the lab. So outside of the host, just in a Petri plate, how well do these secretions inhibit BD growth? Okay, so just to wrap up, hopefully we learned some cool facts about the intricate BD frog study system, and then we zoomed in to learn more about how BD has impacted amphibians in Panama. And then we discussed my research so far looking at the role of skin secretions in the Panamanian golden frog specifically. And with that, I just want to give a huge thanks to the Voiles Lab for all of their support and help this first year, and also want to thank my funding. And with that, I'd be happy to take any questions.
Thank you very much, Jordan. Now we know more about the frog culture and really how devastating this situation is, unfortunately.
Yeah.
So I want to get into some of the skin secretions and just give people kind of a picture of what that might look like in other animals so we can understand on a broader level. What other species are there that have skin secretions, and what kinds of purposes are they used for?
Yeah, so like that figure I was showing, skin secretions are actually found in lots of different animals, in lots of mammals, and in fish. And it usually is for the same purpose of defense against pathogens. So the skin secretions technically are made up of those antimicrobial peptides, which kind of non-specifically target things like viruses and bacteria. So they're often found in the skin of animals, but also potentially in like the mouths or on the eyes of animals. And usually my understanding is it's as pathogen defense.
Mm-hmm. So we see this all over the animal kingdom, of course. And
Yeah.
I'm wondering, on the positive side of the research, are scientists attempting to figure out which skin secretions are effective against PD and maybe figure out how to treat the frogs that don't have the immunity? Or what's the plan?
Yeah, that's actually a big area of research, is looking at frogs that are more resistant to BD, and looking at their antimicrobial peptides specifically and analyzing which peptides are more effective. And I know there's been some work trying to um, like apply those peptides to susceptible frogs, and also similar work looking at the microbiome to see which elements of the microbiome are more effective at inhibiting BD, and then trying to implant those bacteria onto susceptible frogs. And I think the success rate has been quite varied with that, because it's very hard to just add something to a frog when it hasn't evolved with those peptides or with those bacteria. It just won't accept it. So I don't think that's been super effective, but it's been a clear avenue of research for, yeah, conservation medicine.
Mm-hmm. Interesting. And can you talk to us a little bit about what the difference in the antimicrobial peptides is between different frog species? Do they have more of a certain chemical, more secretion in general? What does that look like?
Yeah, I think probably a bit of both. So it is different chemicals, and certain of the chemicals are more effective against BD specifically. And then I think some frogs that are more resistant just have a bunch more. So there's some evidence that maybe larger frogs may be more resistant, and that could be because of more secretions, but it's just so varied. And I think part of what makes this system interesting, but also really complex, is how many frogs are infected. So that's what, yeah, when you're looking at a resistant frog, there's just so many different resistant frogs, and they're all so different. So it's like, what's the commonality that makes them resistant is really complicated to parse apart. So
Mm-hmm. And can you talk to us about what an outbreak really looks like? How does it start? How does it move from frog to frog? And then how long does it take to kind of move its way through the population?
yeah, so like in Panama specifically, it moved pretty quickly. So we can see, and scientists knew it was coming in a way, like it entered above in Central America, it then moved through Costa Rica. So there was some insight that it would be moving down through Panama. And it's mostly, since it's an environmental fungus, probably transmitted usually through humans, these wide areas. And that's the global distribution, likely through the pet trade, through the distribution of these resistant species like bullfrogs into other areas. But down through Panama, it can move smaller distances just through the environment. So it's not entirely determined how it moves, but we know that it can exist in the water and also in the soil. And because many amphibians are so dependent on water for their life cycle that can explain how they can become infected. And then
Mm.
once an outbreak occurs, I know in Panama it was pretty detrimental, like pretty quickly, not many years from seeing dozens to hundreds of frogs around a stream to the kind of dead bodies that I showed in those photos to then just seeing nothing and hearing nothing. But luckily now they're seeing some recovery, but that recovery is really sporadic and their numbers are not near where they were pre-outbreak.
Interesting. Fascinating to study something that moves so fast and is so unknown.
Yeah, and that's what's interesting is at least scientists had some warning of this pathogen in Panama, and actually the frogs I study, the golden frogs, were taken from the environment before the fungus arrived, and now the majority of those frogs that are existing in captive breeding programs. But it was kind of that prior action that really saved them from this infection.
Interesting. So it's just like you might see a larger mammal in a zoo that's protected. You can do the same thing with amphibians.
Right, exactly.
Interesting. So you talked to us a little bit about how you found this specific area of research and why you're so passionate. I wonder for the students who are watching, if you could talk a little bit about maybe someone who doesn't know what they want to study but is interested in science. What would you recommend that they start doing with their education to find their passion?
Yeah, that was me. I mean, I started undergrad with undeclared as a major. So I guess I would just say to really branch out and especially early in your education, just try as many different things as possible. And I know I talked about the dengue virus lab I worked in, but I actually volunteered in 2 other labs as well. And I signed up for another field course that I didn't talk about. So I would say just take advantage of as many opportunities as you can, even if it doesn't seem immediately like this is what I might do or this might be relevant. I would just try and get feelers out and be willing to volunteer is always good.
Hmm. Thank you for sharing. And so I'm curious, I know a lot of your work is done in a lab, but someone like you working on frogs that obviously come from Panama, how does that workload balance? Are you sometimes, of course, outside of coronavirus, are you sometimes out in the field collecting species or working with other scientists and then going back to the lab?
Yeah, that's actually what drew me partially to the Voiles Lab is they have such a connection with Panama. They have those established field sites. So I just started in the fall, so it's been the pandemic, so nobody's been doing field work, but um, and there's definite pluses to doing work in the lab. I really enjoy doing lab work, but I would love to go to Panama and compare the skin secretions of those frogs that have still survived and see if they're different from the frogs I've worked with, which have now been in captive breeding programs. And just in general, it's a really amazing experience to get to go out into the field.
Hmm. So is there a population of Panamanian golden frogs that live there at UNR in your lab?
Yes, yeah.
That's
It's
very
yeah, it's a short-lived population. We just got them this year, and we have about 150 now after the experiment. So we still have quite a bit. I'm going to see them today, but they're very cute. I should mention they're quite small. They're like a few inches, so I really enjoy working with them.
wow. And so will you all do captive breeding as well?
No, the captive breeding is kind of highly regulated to only certain zoos have the, I guess, permits or training to do that captive breeding. So we won't maintain this population. Once these frogs are done, then we just won't have them anymore. But there are several healthy populations I know in Maryland Zoo, and we got some from Nebraska. So luckily, this population or this species has not gone extinct, and it still has healthy numbers in captivity.
Great. And what's the lifespan of a healthy golden frog?
I'm not too sure actually. I think definitely more than a decade.
Wow.
But yeah, I'll have to look into it. Yeah.
Wow, that's quite a long time for a little guy.
Yeah.
Well, we're going to wrap up, but I just wanted to ask you a fun question. What's your favorite species of frog?
I feel like I'm extremely biased, but I have to say the Panamanian golden frog, it's been a real pleasure getting to work so closely with them and I just think they're so darn cute.
Hmm. Great. Well, Jordan, thank you so much for sharing. Always great to have more information about frogs. And if you've been watching this series, you can kind of put these talks together. Some great scientists covering really important conservation work. And Jordan, thank you so much. I hope you have a good day.
Thanks, you too.